This lesson we'll take a look at the macromolecule lipids. Lipids are made up of carbon, hydrogen, and oxygen atoms. They're hydrophobic molecules, meaning that they are non-polar, meaning that they will not dissolve in water, and they can be made up of chains or rings. So lipids have a multitude of structures and functions. So let's take a look at the major structures and functions. The first structure that we're going to take a look at are triglycerides. Tri implying that there are three parts. They are used primarily for energy storage and insulation in organisms. The second type are phospholipids. They form cell membranes and are selectively permeable, which makes sense seeing as things need to get both into and out of cells. Steroids are used for hormonal signaling around the body, cell response to environment and growth. It's basically a means of communication within an organism. And waxes are water resistant and protection, generally found on the outer layers of organisms. So let's take a closer look at each of these types of lipids. So when we look at triglycerides, as the name implies, there are three fatty acids with a glycerol backbone. So when we want to kind of draw a simplistic version, we can draw glycerol with three fatty acids attached to it. However, when we want to draw an actual structural diagram, we want to know what atoms are where. So let's take a quick little look at how we would actually go about drawing a triglyceride. To draw a triglyceride, we start by making the glycerol backbone, which is three carbons covalently bonded together. Those carbons are attached to hydrogens on all sides, except for the side that the fatty acid chains are bonded to. That side has oxygens bound to them. Now, for the triglyceride, it would be attached to a carbon, which is attached to a double bonded oxygen, and then we can simplify the chain by drawing a line like this, because this implies that it is just carbons and hydrogens, which is exactly what a fatty acid chain is made up of. So in the video, we drew the fatty acids as just kind of a zigzag line representing, but this is what it actually looks like. It is carbons bound to hydrogens. So when we want to draw how this macromolecule comes to be, we need to be able to draw the glycerol molecule, three carbons with three hydroxyls. We then need to draw our fatty acid chains. As we did in the previous diagram, we can draw our functional group here, noticing it's a carbon double bound to an oxygen with a hydroxyl group, so we know it is a carboxyl group, and then we draw the rest of the fatty acid chain. We need to draw three of them since it's a triglyceride. Just as our other dehydration synthesis, it's going to be the two hydroxyl groups that are interacting. It's going to be the hydroxyl from one and the hydrogen from the other that are going to leave and cleave off as water, leaving one oxygen left. That oxygen will then bind those two carbons together. That will happen for each of the three triglycerides. And for each bond that's made, notice there are for each bond that is made, there is one water molecule, so a total of three water molecules produced. This type of bond is known as an ester linkage. You need to be able to draw this, so please make sure in your notes you can draw the glycerol backbone, the three fatty acids, and then the formalized triglyceride with the ester linkages. There are two major types of triglycerides, depending on how those carbons are bonded together in the fatty acid tail. There are saturated and unsaturated. As the name saturated implies, meaning it is full. It has the most number of atoms possible. So when we look at this fatty acid tail, it's all single bonds between the carbons, meaning that each carbon has two hydrogens attached to it. These types of triglycerides are solids at room temperature because these fatty acid tails are long and flat and linear. So therefore, when you have two of them together, they can get very close and you can have bonding between them that can hold them together and hold them in a solid structure. You will find these in animal fats like cheese, butter, and things like that. So there are no double bonds between the carbon atoms, which makes for a long linear fatty acid chain. Unsaturated, on the other hand, is not full of hydrogens. It's because there are one or more double bonds within that fatty acid tail. So wherever there's a double bond, there will be a kink or a bending in that chain. So when those chains bend, they can't get as close to each other. Therefore, they can't be as tightly bound, so we often find these unsaturated fatty acids as liquids at room temperature. These are primarily fats that are coming from plants, such as corns, nuts, avocado, and things like that. Our unsaturated triglycerides are generally considered our healthy fats, and saturated are unhealthy. Why? Again, because our saturated are long and linear, they can stick together and they can actually end up clogging up arteries and things like that, causing future health problems. Always have to throw in a cheesy joke to help us make sure that we understand what's going on. The second type of lipids that we want to take a look at are phospholipids. So again, looks very similar, starting with our glycerol backbone. We have two fatty acids this time and a phosphate, as the name implies. So it's a glycerol bonded to two fatty acids with a phosphate group. What's unique about phospholipids is they are what are called amphipathic, meaning that they have both a polar and a nonpolar section. So when we look at a phospholipid, they have a head and a tail section. Those two fatty acids are the tails, and the phosphate is the head. The heads are hydrophilic or water loving, and the tails are hydrophobic, water hating, meaning that they are non-polar. So if we actually look at what it looks like structurally, we can see that there's this phosphate group in the middle. We have this glycerol backbone here, the 3-carbon chain. Notice our ester linkages to the fatty acid tails. You do not need to be able to draw this entire structure, but you do need to know that it's two fatty acid tails, ester bonded to the glycerol backbone with a phosphate group up top. So again, noticing the ester linkages in the phospholipid. These are making up our phospholipid bilayer or our cell membrane, and because of their amphipathic characteristics are selectively permeable, letting some things through, not letting other things through, depending on their polarity or their charge. Third major type of lipids are steroids. What we notice about the steroids is now we start seeing the ringed structure. That defines a steroid. So they consist of 4 carbon rings, in fact, with varying functional groups attached. So whenever you want to identify a steroid, you're always going to see these 4 carbon rings, and you're going to see different functional groups. That's what makes them be able to do different things and interact differently and send different messages. So our steroids include things like cholesterol and our sex hormones. If we take a look at a couple examples, such as the cholesterol, testosterone, estrogen, progesterone, noticing all of them have the 4 rings, but with different functional groups attached at different locations, making them capable of carrying and communicating different messages within the body. And lastly, we have waxes. 3 sections to any wax. There's a fatty acid section, the ester linkage, and an alcohol section. The chain that's attached to the oxygen side of this ester linkage is our alcohol section. So it's a long fatty acid chain linked to alcohol, or sometimes you can see carbon rings. As we said earlier, their job is to keep water from passing and act as a protective layer. We see that on the outside of a lot of fruits and vegetables, which allows them to keep the water on the inside and not shrivel up and get dehydrated if there's a drought, if there's not a lot of rain, keep the moisture on the inside of that organism.